Alright, so our last lecture we left with motor neurons or somatic motor system. And what are those going to control? Those are going to be your skeletal muscles. So chiefly our lecture today is going to be on skeletal muscle. Um, we do have two other types. We got the smooth muscle and cardiac muscle, which we'll touch on briefly. We'll get into a lot more of the cardiac muscle when we get to the cardiovascular system. But today we're going to chiefly talk on skeletal muscle, and then we will touch just briefly on the other two. All right, so like I said, there's three different types of muscle. We got the skeletal muscle, the cardiac muscle, and the smooth muscle. We're going to chiefly talk on the skeletal muscle today, but we will touch on the other two. So let's just look at some of the basic function and basic characteristics of these muscles to get us going. For our skeletal muscle, if you remember, they are attached to the bones and they transverse a joint. So when they contract, they are controlling body movement. They're making movement of our bones. And also, if you remember, these skeletal muscles are controlled by the somatic motor nervous system. So that's our voluntary motor system. We have to consciously think about contracting our muscles. Okay, the cardiac muscle. Okay, the cardiac muscle is going to be making those walls of the heart. And so when it contracts, it's going to provide the force to push the blood through the bloodstream, pushing into the blood vessels and throughout the body, or throughout the blood vessels throughout the body. Right, and we'll look skeletal muscle, cardiac muscle, as you see, are striated. We'll talk about that in just a second. Nervous system wise, cardiac muscle is controlled by the autonomic nervous system. And that's our involuntary nervous system. So we don't have to consciously put thought into contracting our muscles, our cardiac muscles. We don't have to consciously think about the beating of our heart because it is controlled by the autonomic nervous system. Smooth muscle, you'll find this mainly around internal organs and the tubes. A classic example of smooth muscle is smooth muscle wraps around the blood vessels. Blood vessels is a tube. Smooth muscle wraps around in a circle around it, and when it contracts, it will control the diameter of the blood vessel. The diameter is larger, vasodilated, or when it's contracted, it will be vasoconstriction, and the diameter of the blood vessel will be smaller. Just like the cardiac, smooth muscle is controlled by the A and S, the autonomic nervous system, because we don't have to consciously think about controlling our blood vessels. What's the diameter of our blood vessels? Okay, our automatically does that for us. So now let's look anatomically some of the similarities and differences. We can look at skeletal muscle. We already talked on striations. Those are these lines that you see in both the cardiac and the skeletal muscle. So cardiac and skeletal muscle. And that means these two muscles have a similar arrangement. The proteins arrangement in these muscles is similar. They have a similar setup. Whereas a smooth muscle, you don't see those striations. Okay? So it has a different arrangement than that of the cardiac or the skeletal muscle. Some differences between, say, skeletal and cardiac is you will see in the skeletal over here, we will find they are multinucleated. Okay? The muscle cells, or what we term muscle fibers, are multinucleated. And these muscle fibers, kind of like a neuron, they can be microscopic in length, but the muscle cells run the whole length of the skeletal muscle, so these cells can be up to several feet long. And so they need the lots of proteins and so forth, so it's key that they are multinucleated. Okay, if we jump down to the cardiac muscle, just like the skeletal, has the striations, but you can see these are very smaller cells. You can see here's one end, here's another end, and there should be another end here. But these are small in length, and they are also branched. And then the other telltale sign that the skeletal muscle doesn't have is the cardiac has these intercalated discs where you have the meeting between the two cells. And we'll get into the significance of this intercalated disc when we get to cardiovascular systems, because we will talk more on the cardiac muscle. Right, so that's kind of the basic similarities, characteristics, anatomically, and also control of them, and allows us to get us to moving on further. But again, we're going to focus chiefly on the skeletal muscle. So when we start talking about the skeletal muscle, or the muscles, we got some special terminology for these cells. Just a general here. A muscle cell, so when you hear me say muscle fiber is a muscle cell. The cell membrane for a muscle has a special name, sarcolemma. Okay, you'll see the sarco for the prefix for most of this. The cytoplasm is the sarcoplasm, and then there is a modified endoplasmic reticulum that is important for the operation of the muscle, and that is the sarcoplasmic reticulum. Okay, so you'll see these names used throughout this lecture. These are special, significant to the muscle cells. Okay, but they are these components of your basic cells. Okay. So here's the basic setup of the muscle. Okay. If you remember our muscles, they are attached by tendons to the bones, right, and they run across, but they have to traverse a joint, okay, and connect it to another bone with a tendon. Okay, and we will see with muscles, the only force they can produce is a pulling force, because all they can do is contract. All they can do is shorten. There's no force when the muscle is lengthening. The only force is the contraction force as it gets smaller. Okay, but as this muscle, you can see as it gets smaller, it's going to pull this arm towards the upper, okay, forearm towards the upper arm. So that's just how these skeletal muscles operate. We're basically forming a lever system. Okay. And you can see this is a whole muscle, and this whole muscle is going to have many muscle fibers, but those muscle fibers run the whole length. So you can see how these muscle cells are quite long. Okay. And so like I said, muscles only produce a pulling force. There is no force when the muscle is lengthening. So the force comes from the muscle shortening. Okay. So that requires that there are antagonistic muscles. Okay. To flex the arm, I need a muscle on the front here that pulls the arm towards shortening. Okay, but this muscle has no force pushing the arm back. So how do we do that? We need an antagonistic muscle, and that would be the triceps. This is the posterior portion of the arm. Okay, it's going to pull this way. Okay, so that's how we get the different movements, is we got to have antagonistic muscles, because the muscles can only pull. There is no pushing force in the muscle. Alright, so let's look at the basic anatomy of a skeletal muscle. Okay, here is a whole skeletal muscle, and you can see it is arranged with a multitude of muscle fibers. So many muscle fibers are going to make up a whole muscle. Okay, so you have down here these individual muscle fibers, and these individual muscle fibers are grouped, are bundled together in what we call a muscle fascicle. So just a bundle of fibers, individual fibers, makes a muscle fascicle, and then you can see when we look at the whole muscle, the muscle fascicle here, there's another fascicle here, and another fascicle here. So a bundle of muscle fascicles can make a whole muscle. Okay. And as you can look, you can kind of see there is connective tissue component to the muscle. So it's not just contractile muscular component, there is also a connective tissue component of the muscle. Okay. And the endomysium, okay, is a connective tissue layer that wraps around each individual muscle fiber. Okay. Then there is a perimysium, in which wraps around the fascicle. Okay, you can kind of see the perimysium located in here, the white. All right, and then there is the epimysium, is the connective tissue component that wraps around the whole skeletal muscle. Okay. And at the end of the muscle, that epimysium binds together to become the tendon, which is going to connect the skeletal muscle to the bones. Okay, so that's the tissue component, or sorry, connective tissue component of the muscle. So there is both contractile tissue, that's the muscles, the muscle fibers, and then the connective tissue component that gives muscle some of its elasticity. So looking at an individual muscle fiber here, so this is a blown up of that smallest component we saw in the previous slide. So this is a cutout of a blown up portion of an individual muscle fiber, so just one muscle cell. Okay. And in here we're going to see what are kind of the main structure of a muscle is the myofibrils. This is this rod-shaped organelle looking here. Okay, and there's multiple of these myofibrils throughout a whole muscle. Okay, and these are going to have the contractile and elastic components of the muscle. This is where the contraction or the shortening is actually going to take place. You'll see we'll talk about these thick filaments and thin filaments, all right, and that's just basically the arrangement of the myofibril. Also, there's that modified endoplasmic reticulum called the sarcoplasmic reticulum. Okay, that modified, and that is this blue component. You can see it wrapping around, okay, associated with these myofibrils. And we'll talk a lot about the sarcoplasmic reticulum because it has a special function that's going to make these filaments in the myofibril to cause shortening and causing our contraction. So now looking from that previous slide, if we blew up a region there, right, it'll get us something looking like this. And one thing we want to note, here's the sarcolemma, so that's the plasma membrane of the muscle fiber. And we're going to see to make this muscle contract, we're going to have to send an action potential. And an action potential has to get to all of this region of the muscle to make sure we get contraction. Okay, so if the action potential is coming along the sarcolemma, we need to get that action potential deeper into the muscle. And that's what this arrangement, this transverse tubule, will be doing. It's basically, you can see, just an invagination of sarcolemma, the plasma membrane. Okay, so that's can bring that action potential deeper into the muscle here. So you can see it. This is going to bring the action potential deeper, and you can see these blue sacs. We'll talk about these in a second. Those are those sarcoplasmic reticulum. Okay, so basically that T-tubule is to rapidly move in the action potential deep into the fiber. So we get the action potential running through all of the muscle so we can get contraction. Because we're going to see later, the action potential is the cue to get the shortening of the muscle. Now looking closer at the blue portion, all right, there's this region here. This is the sarcoplasmic reticulum, this blue, all right, and it ends at this terminal cisternae, this enlarged region, right? And it is this terminal cisternae is associated with that T-tubule there. Okay, so the action potential is coming down here, and we have interaction between the terminal cisternae, okay, of the sarcoplasmic reticulum. Okay. And we're going to see what's the function is that sarcoplasmic reticulum. You can think of it as like a big sack of calcium ions. It's holding in calcium ion. It has a high concentration of calcium ions inside here. Okay. And we're going to see we release those calcium ions, then they can go out into this sarcoplasm, okay, where the contractile opponents are. But basically think of these sarcoplasmic reticulums as big sacks of calcium ions. So now we're going to get even deeper into the muscle. We're magnifying even more, and we're looking at, this is a single myofibril. And remember, there'll be many of these rod-shaped organelles running down the length of a muscle fiber. Okay, and so we're going to see this arrangement. Okay, and it's arranged in a sarcomere. Okay, and these myofibrils are going to contain the contractile components of the muscle. You're going to see here is, if we took out, so here's the edge of one sarcomere, and here's the edge of one sarcomere. And you can see running down the whole muscle, it's just a repeating structure of sarcomeres all the way down the muscle. Remember, this myofibril will run the length of the muscle, so it can run several feet. Okay, so you'll have these multiple repeatings of sarcomeres down the length. But let's take a look at each of these individual sarcomeres. Okay. You're going to see it's made up of two components. For the contractile components, we got the thick filaments, which are the myosin, and the thin filaments, which are the actin. And you can see the arrangement. We're going to talk in detail about this, but it's arranged in this sarcomere. Okay. And then the thick filaments that you see here, they are composed of these myosin molecules. Okay, hundreds of myosin molecules that have this tail region and then a head region with two myosin heads. Okay. And you can kind of see the arrangement of these myosins here when we look at the filaments. Is one side, okay, if the M line's the middle of the setup, you can see one side the heads are facing this direction, where the other side the heads are facing this direction. And we're going to see what happens is this head is going to swivel this direction. So it'll end up being more like this. So it's going to contract. Okay, it's going to do a stroke. then movement is this way. Okay, so these heads would be moving this way when it's stroked, and these heads would be moving in this direction when it strokes. Okay, and then we'll talk about, we see the thin filaments. Those are the actin. Okay, and what we're going to have is an interaction between the thick filaments and the thin filaments, which is going to allow us to do the contraction. And so we'll get into that in just a sec here. All right, so looking again at the sarcomere, right? That's this whole thing here. All right, and this is the basic functional unit of contraction. This is where the contraction of the muscle is going to take place. All right, and we have proteins that are involved in contractile proteins. Those are the ones that are actually going to cause the shortening, to cause the contraction. We got proteins that help regulate whether the muscle is contracting or not, and then some accessory proteins. So we've already taken a look here. That is the myosin head. So this is part of the contractile. That's this here. The pink is the actin. That's the thin filament. Okay, you can see with all those actin molecules. All right, and then we have regulatory proteins. This is going to control whether the muscle's contracting or relaxing. All right, and those are down here on the actin, the troponin and the tropomyosin. Okay, and then some accessory proteins. These help keep the structure of these thick and thin filaments. You'll see tintin is here. This is this green elastic protein. This gives the elastic property of the muscle, and also anchors this thick filament. And then down here you can see our nebulin. That's okay. The nebulin is this guy here, right through the middle, and it gives the structure or the bones for the actin filaments. Okay. So let's first look at thick filaments. We already saw this is part of the contractile or motor proteins. This is what's going to cause the contraction to take place. And we saw this myosin molecule has a tail region and then has the head region. And you can see there are 100 of these arranged, okay, to make the thick filaments of the sarcomere. Okay, so 100 of these molecules, you can see there's 1, 2, make up. But again, you see the arrangement of the heads. This side of the thick filament, the heads are running this direction, and this side, the thick filaments are running in this direction. And like I said, that myosin head is going to swivel this direction. Okay. So this one, these heads will swivel in this direction, pulling this way, and these heads will swivel in this direction, okay, pulling that way. So as for the thin filament, the actin, okay, the actin is made up of these individual little G-actin units, these little balls, okay, that are connected together. All right, so we got these long strands of these G-actins that make what's called the long strand is the F-actin. All right, and then you have two strands of F-actin, basically wound up, kind of like the DNA. It's in a helix, rounded up. Okay, and so that's what makes up the F-actin. Okay, long strands, two F-actins make up the actin molecule, or sorry, the thin filaments. Right, and you'll see on this actin, we're going to talk about another protein, troponin, and then there is this green protein, tropomyosin. Okay, and on this actin, you can see there are two different colors. There's this reddish color and then this yellow color. This yellow color is basically the active site. This is where our myosin heads, where the myosin heads from the thick filament are going to interact with the actin, right? And this is going to be the modes in which we do contraction of the muscle. So we get an interaction between the thick and the thin filaments, but the sites are, this little yellow area is where the myosin would bind. So now that we talked about the thick and the thin filaments, you can see how the arrangement is again in the sarcomere with the thick filaments here and the thin filaments running there. And what's going to get us, allow us to contract or do what the muscle is going to do is to contract, to shorten, is going to be occurring by an interaction between the myosin head and the actin molecules. And we get what's called cross-bridging. So we get this interaction here. This is the cross-bridging between myosin and actin. And you can see, remember the heads. The head in its relaxed state is here. Okay, and it's going to move from here to this direction. Okay, so if this head was to come on and bind to this here and we get a cross bridging and then the head moves this direction, what's going to happen to the actin? It's going to be pulled in this direction. Okay, so here bind, boom, pull it this way, it gets pulled that direction. Okay, so where we're looking at here, you would find the setup right here, because the heads, you can see the myosin heads are arranged in this order. Okay, so this head binds, power strokes, and then that moves the actin this direction. This head here would move and power stroke that way and would move the actin in this direction, because you can see how this cross bridging, this interaction along with that power stroke, moves this side of the sarcomere this way, this side this way, and if that takes place, sarcomere total length at the end will be maybe something in that, much shorter than what we see down here. Okay, so that's going to be our gist of how we get shortening and getting contraction of the muscle, is through this cross bridging and along with this power stroking of the head. So let's just take a look, also closer look at those accessory proteins, and these are basically, this gives it some of its elastic property. This tentin, okay, this tentin here provides the elasticity. It anchors this thick filament, but it is also elastic because your muscles, all right, what happens, you can stretch your muscles, right? And what are you stretching? You're stretching mainly this elastic component of the sarcomere along with the connective tissue fibers in the muscle. Okay, but that gives the muscle some elasticity. Okay, what would happen if it stretches too far, right? You can break this, but this tentin is this elastic molecule, helps stabilize and also allows stretch of the muscle so it's not rigid. Okay, this is actually the largest protein in your body. Nebulin. Nebulin is a little blue running along here. Okay, and that helps align the actin, and it connects to this Z disc here to help align the actin molecule in the sarcomere. Okay, so tentin, elastic, helps align the myosin, and the nebulin, inelastic, but it helps align this actin in the sarcomere. So let's get down to looking at what this actual functional unit of contraction Okay, we have, here is our sarcomere, and you can see both, this would be one full sarcomere here. It's flanked on each end is the Z disc to help anchor those thin filaments you saw earlier. Okay, and it's an attachment site for those thin filaments. Okay, you got one on both sides of the sarcomere. So those are the borders of each sarcomere. And if we're looking, we can see what's called I-band. Okay, and you can see the I-band here, where we see those striations in the muscle. We see a light and then a darker color. That gives us our striations. Okay, that lighter color is the I-band. Okay, and you can see the I-band here. What filaments are in the I-band? Okay, the I-band is solely made up of thin filaments. Okay, the I-band contains thin filaments from one sarcomere and the neighboring sarcomere, or thin filaments from a neighboring sarcomere, because this is the Z disc. This is a separation between these two sarcomeres. Okay, so thin filament makes our I band. Okay, and then you can see an A band. That would be this right here. Okay, and what filaments do you see in the A band? You see thin and you see thick. Okay, going across thin and thick. So the A band contains both thin and thick, and so it gives it this darker appearance. Okay, looking there. Let's go. There are some more parts. We'll talk about the H zone here. Okay, the H zone, it's the central region of that A band, and you can see the H zone is here, border here, and here. And what do you see? Just solely thick filament. It's only occupied by thick filament. Okay, and then the M line is this middle line. Okay, and attachment site for the thick filaments. Okay, that's kind of the midway point of the sarcomere. Okay, those thick filaments are going out in one direction, and the other ones are going out in the other directions, rather than the heads are two different directions on each side of that M line. So when you look at the sarcomere, it's actually this three-dimensional type structure. It's not this two-dimensional, but it's this arrangement of thick here and thin filaments, and if you put them together, you can see there's this arrangement of thick and thin wrapping around each other. But there's thick has those heads that will attach to there, there, and then these will also attach to there. So we got this arrangement where these thick filaments have interaction with the thin, not just two-dimensional, but three-dimensional. So here's where we've gone so far. We've gone through the anatomy. We've gone from a skeletal muscle and broken it all the way down to those individual molecules that make up the myofibril. Okay, and then these are all arranged in that sarcomere. Okay, and so down the line we have those repeating pattern of sarcomeres. Okay, down those myofibrils. Okay, those rod-shaped organelles. And so we're going to get all these sarcomeres. We get all that myosin interacting with that actin and pulling that actin closer together. Okay. So what are we trying to accomplish with our muscle contraction? Basically, we're trying to create force to do movement. Okay. And to do that, we got to have one phase, the contraction phase. Okay, that's the phase where those myosin heads are running down or moving the actin closer together, and we're going to get shortening of the muscle to create that tension. Okay? And of course, you can imagine we got to do this movement, and that's going to require lots of ATP, lots of energy. Okay? Then we also got to release, right? We don't want our muscle to be tense and just basically in tetanus, just completely contracted all the time. We have to relax it. Okay? And surprisingly, what you also need for relaxation is you will also need ATP. Okay, so you can see this muscles are very energetically, need lots of energy. Okay? So need lots of ATP, need lots of fuel, need lots of glucose or fats to metabolize so we can create that ATP. Okay, and what are we trying to do? That force we're using to move a load, whether it's our own body weight, whether we're at the gym moving weights, but we're creating force to oppose. The load opposes the contraction. The contraction is what's going to move the load. And to accomplish this, we're in a sliding filament theory of contraction. It's a theory of how the filaments actually slide across each other to create contraction. Okay. So if you remember, we got those sarcomeres that are overlapped, the actin and myosin overlap each other where they get interaction between each other. Okay? But those actin and myosins, the key is they are fixed length. They don't change their length. What they do is, as you see down here, they will slide across. Okay, so that myosin is binding here and pulling the actin in this direction, right? And the myosin here are pulling this actin in this direction, and this one's pulling this in this direction. So these actins are moving closer together, but the length of these filaments are not changing. Basically, the actin is sliding across those myosins to move closer to that M line. Okay, so we bring it closer. So remember, the muscle is just a repeating continuation of this, the sarcomeres, all the way down the line. So if all these sarcomeres go from this configuration down to this configuration, that muscle is going to shorten substantially, all right, and we are going to get contraction of the muscle. Okay, but this is the sliding filament theory, is that these filaments are basically sliding, the thin are sliding across the thick, and the thin are coming closer together, and we get contraction. Okay, so again, the filaments aren't changing. What is changing though of the sarcomere? Look at the I band here, and look at the H zone. Okay, if you remember, the I band is consisted of solely thin filament. The H zone consisted of solely thick filament. And so what do you see changing from a relaxed configuration of the sarcomere to a contracted configuration? You can see that what part of the sarcomere is thin now. Just this portion compared to this big portion up here. So the I band gets smaller. What about the H zone? The H zone extended here and here in the relaxed, but now we just have this small, because this is the only region that contains thick in between here. So the I band and the H zone both get smaller when We're doing the sliding filament theory when those filaments slide across one another. Okay, so we kind of got the gist of how the muscle shortens by those sliding filaments. Those filaments are sliding against one another. But let's put it all together. Let's put all the events that have to take place to basically make this muscle contract and how we actually get those filaments to slide across one another. Okay, there's three major events. We got the ones at the neuromuscular junction. Okay, that's going to be with our motor neuron. We got excitation contraction coupling. And we get the contraction relaxation cycle. Okay, so these all three have to take place to give us a successful muscle contraction. We're going to kind of go backwards. I'm going to start first with the contraction relaxation cycle. So kind of what we just touched on, that sliding filament theory occurs through the contraction relaxation cycle. So looking at the contractile cycle, how do we get the myosin heads to interact with the actin first, and then to do that power stroke, to move the actin, to pull the actins towards each other. And you look here, we got the two players, we got the myosin head, and we got the actin. The actin, you can see, has two colored regions here, right? The red and the lighter orangish color. That lighter orangish color is the myosin binding site. That is where the myosin wants to bind. Okay? And we have two regulatory proteins that we've talked about here. We got the first, we have this troponin. This little TN molecule here is troponin. And what its job is going to be is it can bind calcium. This green string-like structure, okay, on both sides, you can see it's covering up these myosin binding sites. It's covered over. Okay, that is tropomyosin. Tropomyosin interacts with troponin. Okay, troponin binds calcium, but it also interacts with tropomyosin. And you can see tropomyosin is covering up that site in which this myosin head can bind. So when tropomyosin is in the way, the myosin head cannot bind. All right? And when troponin is not bound, there's no calcium here. This is how tropomyosin looks. Okay, it will be covering those myosin binding sites. All right? Now, let's get us some calcium. Okay, we'll talk about where calcium comes from later, but let's just put calcium in here. If calcium is available, it's bound now to troponin. Troponin, again, interacts with tropomyosin, and it will move. When calcium is bound to troponin, it moves tropomyosin off those sites, and now those sites are available for our myosin head to interact. Okay, our myosin head is going to interact, and then we get the power stroke. Okay, if we power stroke, we're moving the actin in that direction. We're pulling it. Okay, so that's essentially what is happening. First, we need calcium. We've got to have calcium so we can get the cross-bridging to take place. Okay, and then we can initiate the power stroke. All right, so we've seen how we can get that calcium signal allows us to get interaction between myosin and actin. Now let's look at what fuels that power stroke. So the next two slides talk about the contractile cycle. I'm going to jump 2 slides to get to the picture that actually is showing what is taking place, what these words are saying. Okay, so We get power stroke. We'll talk about this. We'll look at the slide here just next that we'll look at the picture arrangement of this. Okay. So the contractile cycle, basically what is happening? The head is binding, okay, and here it has already done the power stroke, but we need the myosin head to bind power stroke, and after the power stroke we need this head to release so it can go up and bind the next actin molecule and power stroke that way, and then we have to have it release again, bind here, and power stroke, because basically this myosin is walking this way while pulling the actin molecule that way to give us our sliding filament theory. All right, so after the first power stroke, we have to release. So how myosin head releases is we need ATP to bind. To get the myosin head to release, we must have a new molecule of ATP. So ATP must be available to release the myosin head. Myosin head has an ATPase. It has an enzyme that splits ATP into ADP and phosphate. Right, but see here it is still bound. Okay, when this is a high energy state and it allows for binding. So the ATP binding releases the molecule, but splitting causes the molecule to interact again with actin. Right, and of course there is a calcium signal here that is allowing the ability of myosin and actin to bind to each other. Okay, now what do we see taking place from here to here is when the power stroke takes place, the phosphate is released. Okay, but ADP is still bound. And then ADP releases. Okay, so those are happening very quickly, and now the myosin is attached to the actin. Okay, and it will stay attached until we get a new molecule of ATP to release it. So we are doing this just over and over and over again to move those actin molecules, to move them, to pull them towards the M line. All right, so we've seen, we have three separate events to get our muscle contraction. We've seen the contraction cycle. Okay, we'll talk about the relaxation part basically at the end here, at the very end. Okay, but we're going to jump up here to the events at the neuromuscular junction. So we're going to be talking about neuro. We're going to be talking about the neuron that talks to the muscle. So this probably looks familiar from your anatomy days, but the neuromuscular junction, because this is a classic synapse that we talk about in anatomy. But you have that somatic motor neuron, and it is talking to the muscle cell here. Okay, so here is the neuron running in. Okay, little telodendrias, and then we get synapsing with the muscle. And here's a connection, and here's kind of what it looks like at the synapse. Okay, so what do we have here? We got the presynaptic neuron. Right, this is the synaptic nerve or synaptic terminal. Okay, and we got those vesicles. In this case, we got acetylcholine. We got the synaptic cleft. That's just the space because there is not a physical connection between the presynaptic neuron and the muscle. And this is the membrane of the muscle. Okay, the sarcolemma. And this is the motor end plate. It's a specialized arrangement because it has these invaginations and it has a high density of these receptors. Okay, those are your nicotinic receptors. Okay, those are ligand-gated ion channels that are going to let sodium in. So if we're letting sodium in, what's happening to the muscle? Yeah, we're getting depolarization. So we got the motor neuron with acetylcholine neurotransmitters and synaptic cleft, the motor end plate of the muscle with those nicotinic receptors. So hopefully this looks familiar from our neuro days, but if you remember, we got our action potential coming down to the terminal, and that action potential causes those calcium, voltage-gated calcium channels to open, causing influx of calcium. And then the influx of calcium causes the release of neurotransmitter. In this case, we're talking acetylcholine. Those somatic motor neurons contain acetylcholine. We get diffusion of acetylcholine, and they are going to bind to the nicotinic receptors that are located on the motor end plate. Okay, and in this case, the main flux of ions that are going to be in, it's sodium, potassium, but mainly sodium. So if sodium is rushing in to the muscle, what's happening to the membrane here? We're getting a increase in membrane potential. All right, we're getting a depolarization, going in the positive direction because all those positive sodium ions are rushing in to depolarize the membrane. So release of neurotransmitter, binding of acetylcholine, and that causes sodium influx, and we get depolarization of the membrane. So just a closer look, you're looking at, here's the actual nicotinic receptor. Okay, it's a ligand-gated ion channel. We get acetylcholine, 2 acetyl molecules binding, and that's going to now allow sodium in and potassium out. But the big influx is sodium. So if we have more positive sodium coming in, we're going to get depolarization of the membrane. All right, so we've seen what's taking place at the neuromuscular junction. We got basically depolarization of the cell, and just very similar like a neuron depolarization, if there is enough to reach threshold, we're going to have an action potential down the sarcolemma of the muscle. So that brings us to the events that take place at the excitation contraction coupling, EC coupling. So the excitation, that's the action potential, and the action potential is going to lead to events that ultimately lead to the muscle contracting. So we're coupling those two events together, the action potential along with what's going to take place to cause the contraction. Okay, that will bring us into that contraction and then relaxation cycle. So we've talked contraction, we'll talk about relaxation in just a little bit, but first let's see what takes place at the EC coupling. All right, so EC coupling, we've had our events here. This is what's taking place, okay, at the neuromuscular junction, and now we have an action potential. We got depolarization, we're moving our action potential. Okay, there are voltage-gated sodium channels, voltage-gated potassium channels, just like a neuron, that propagate the action potential down the membrane. Okay, and if you remember, we got the T tubules that are going to be associated with the terminal cisternae of the sarcoplasmic reticulum. And that sarcoplasmic reticulum is basically a big bag of calcium. Okay, so what do we need for contraction to take place? We got to have calcium and we got to have ATP. So let's get our calcium out there. We need our calcium to bind to troponin to move tropomyosin. All right, so the action potential, is coming down the T tubules. In these T tubules, you have this DHP, okay, dihydropyridine calcium channels. And they are physically coupled to these RYR, which are your ryanodine receptor. Okay, these are basically channels, and when they open these channels, the calcium is going to rush out and flood the sarcoplasm. Okay, and what's out here, right? That's where our troponin and tropomyosin and all our contractile filaments. Okay, because calcium is the cue to make that sliding filament take place, to move, okay, allow the binding between myosin and actin. All right, so the action potential comes down. That action potential is going to activate the DHP, which is physically coupled, which is going to cause a conformational change to open up this ryanodine channel. And now we get calcium just flooding out. Okay, so each time an action potential comes down, we keep opening this channel, more calcium comes out. Okay, so calcium's the cue for our contraction. Okay, and this is how we get it, through this EC coupling, because calcium is going to bind to troponin, move tropomyosin, and now the myosin head can bind to actin. So just a closer look of what's happening at the EC coupling. Right, we have that action potential coming down, and you can see better the cueing of that activates those DHP receptors, which are physically connected to the ryanodine, and that opens up the ryanodine channel. And now we can get calcium rushing out, and that calcium binds to troponin, moves tropomyosin, and now we can get interaction of the myosin with actin. So that's to get contraction. What do you think would we need to do to get relaxation, right? Muscles contracts, now we got to relax it. Okay, and what was the cue to contraction was the calcium signal. So if we're able to remove that calcium signal, that'll allow us to give us relaxation. Okay, so we've seen the calcium cues the contraction. The EC coupling's done. We get the calcium out, and we get the movement of binding of calcium to troponin and the movement of tropomyosin. So how can we get relaxation? We need the muscle to relax. And so what was our cue to contraction was calcium influx, or calcium in the sarcoplasmic. Okay. Now, to cause relaxation, the opposite of contraction, we can remove the calcium signal. If we remove the calcium, then we can get here. Myosin cannot bind to actin. So to do that, we have to actively pump the calcium back into the sarcoplasmic reticulum. Okay, so the calcium is constantly getting pumped back up into the sarcoplasmic reticulum right here. Okay. And now there's no calcium. If calcium's gone from here, we can't bind, and so now the muscle's in relaxation. Okay, so to get contraction, we open up those channels and calcium rushes out, and then to get relaxation, we have to use energy, ATP, to pump the calcium back into the sarcoplasmic reticulum. So which two of those events do you think is faster? Opening the channel and calcium rushing out, or having calcium being pumped back into the sarcoplasm? Yeah, the rushing out is much quicker. So we'll see what's taking place in our muscle twitch. We have our muscle twitch down here. We have a contraction phase, okay, and then we have a relaxation phase. Which one looks like it is more drawn out on time? Okay, the relaxation phase is basically almost twice as long as our contraction phase here. Okay? Because here, calcium's channels are opening, we're getting rush into calcium, we get rush of calcium, right? And then we get contraction, and then we get pumping of, and then the slow pumping back, removing the calcium, and then we slowly get relaxation. Okay, there's another phase here before we actually see contraction, this latent phase. Okay, and that's where the EC coupling is taking place. Okay, so all that action potential stuff is taking place here before we actually see the muscles contracting. All right, so let's kind of put together the electrical and mechanical mechanisms of making a muscle contract. And what's causing muscle contract, right? We got an action potential, and we could manually do an action potential by basically activating a muscle with an electrical impulse. Okay, but if we did that once, we would get a muscle twitch. So a twitch is a single contraction with relaxation cycle. So you can see here we get contraction phase, and then we get a relaxation phase. What's off about the relaxation phase here is this should be more drawn out, right? Because this is the slower phase. One other event I want to show is this latent period. Okay, because this latent period, if you can see, here is the action potential that's running down the muscle. Okay, this is going to become important later when we start stimulating it quicker and quicker. But with one twitch, we get the action potential running down, and what do you see? You don't even see, time-wise, the contraction starting to take place, the actual force, till after the action potential's already finished. Okay. So let's look at muscle twitch. Okay, as we said, there's three main phases: the latent period, and what's taking place through that latent period. Those are all those EC coupling events, right? Because we activated it, we sent an electrical impulse, we have an action potential, but what's that action potential? We got to move the action potential down the transverse tubule, open up the channels, get calcium out. We got to have calcium bind to troponin and move tropomyosin. Now we can actually start the contraction phase. So we have all those events in the EC coupling taking place that take time before we actually see contraction. Okay? Then we get contraction, and then what's causing our relaxation? Yeah, remember we're pumping back that calcium back into the sarcoplasmic reticulum. But again, this should be a longer phase. This should be longer drawn out because it's double that of the contraction. So this graph is a little bit off here. It should probably look more like how I'm drawing it here. Okay. What determines how fast and how this rises? It has to do with some of the speeds of the fibers, okay, and tension. Some can create greater tension, and also the duration. Some can, when our fast twitch, they go up very quickly and down very quickly. So they got a fast contraction phase and fast relaxation phase. Okay? But that is a single muscle twitch. We got a latent period, the EC coupling. We got the contraction phase, and then follow that with the relaxation phase for our simple muscle twitch. Now let's start activating these quicker and quicker. If we were to start, we only shocked our muscle once. We gave it one electrical impulse and caused one muscle twitch. What would happen if we started decreasing the interval between action potentials? If we shocked it multiple times continuously, okay, you'll start getting what we see as summation. Okay, because what happens every time an action potential comes is we open up those calcium channels, right? Every time an action potential comes down, we're opening up those calcium channels, more calcium can rush out of the sarcoplasm. Okay, and so think about it, if we had more calcium, could we move away more tropomyosin on all those myofibrils in our muscle?
Yeah, we could. So if we moved away more calcium, we can also get more myosins binding to actin. And what do you think that would happen to tension if we were able to bind more myosin heads to actin? More heads would cause more tension. So as we increase the stimulation, okay, we stimulate the muscle more and more quickly, we're getting more calcium out into the sarcoplasm, and we'll start getting what we see as summation. Okay, and then if we start going higher frequency of stimulating, we're shock the muscle, we can get what's called unfused tetanus, and you can see the tension will rise and the muscle fibers are continually to rise, but you'll see they don't come back down to full relaxation. So we're shocking the muscle before it can even relax again. So we keep on opening up those calcium channels. All right, and then if we continue to even increase the stimulation more and more, we can get what's called complete or fused tetanus. Okay, that's where the muscle fiber has no time to relax, doesn't have any time to let the calcium get pumped back into the sarcoplasmic reticulum. All right, so let's take a look at these. You can see here's a muscle twitch. We shock it, goes up, comes down, but it has time to relax. If we start increasing, here's a stimulation, here's a stimulation. Okay, before we stimulated here and here, okay, we had time for the muscle to relax. Now we're starting not to give the muscle time to relax, and we can start increasing. If we increase calcium, right, if calcium never gets fully pumped back, we can't get relaxation. Okay, so now we start getting summation, and now here's shock, and more calcium. Not enough time to pump it back. We'll increase the calcium, and we're getting very little time for relaxation. Okay, and as you can see from before here, the tension also is increasing. This tension is much greater than what we see up here with the single muscle twitch. And then if we shock even more, okay, we'll go from this unfused tetanus to this fused or complete tetanus. Okay, this is a little off. This should actually show maximum tension. Should be further up here compared to this, but this would give you your max tension. All right, but what's happening is we're shocking it, we're stimulating it so fast that it doesn't have time to relax. Okay, so give us this fused tension. So why can we get this ramping up, this tetanus of the muscle? How can we get it to maximum tension and to keep it from relaxation? And that kind of goes towards the difference between timing of the actual action potential compared to the actual muscle contraction. Okay, if you look, here is the action potential in the green, circled in the green, this event here. And just like a neuron's action potential, there's an absolute refractory period. But that absolute refractory period is ended here. What's happening contraction-wise in the muscle? The muscle hasn't even started contracting before we can send the muscle another action potential. So I can send it another action potential here, probably another one here, and maybe even another action potential here. And each time we send that action potential, we keep opening those gates to let calcium out. And where do we get to relaxation, right? We've basically hit it with open the gates 4 times now before we even try to get to relaxation. So because of that short refractory period, we're allowed to stimulate the muscle multiple times before we get to relaxation or contraction, and therefore we can ramp it up. And then we get this fused tetanus, this complete, basically, tension of the muscle. Now, will we want to do this with our cardiac muscle? Because we'll be talking about this later with cardiac. Yeah, do we want tetanus? Do you want your heart to be in complete tetanus? No, and we're going to see there's a reason why for this, because this action potential will look different in the cardiac muscle, but this is a short refractory period, and therefore we can stimulate it multiple times before we even get the mechanical events taking place in the muscle. So now we've looked at the properties of contraction. Let's look at type of contractions that can take place. Two basic types. You got an isotonic contraction and an isometric contraction. Isotonic, classic, is when I go to the gym, I'll do my bicep curls, right? I'm bringing that muscle up, I'm moving it towards my arm, I'm generating movement. That's an isotonic, because what's the shape or the size of the muscle is changing when I create or generate the load, generate the movement, okay? Whereas isometric, I'm still getting contraction, but I'm not moving the load. So if I go in and pick up an 80-pound dumbbell, it's too heavy for my muscle to resist the load. So I can't bring the dumbbell up, but my arm is still contracting, my muscle is contracting, but what's not happening? I'm not creating movement, I'm not changing the shape or the size of the muscle, okay? It's staying put. I'm not generating a movement. That's an isometric. So isometric, same metric. I'm not changing the length of the muscle, whereas when I'm doing an isotonic contraction, I'm changing the length. In isotonic, I can have two different movements. I can have an eccentric and a concentric. When I go to lift that dumbbell up, all right, bring it closer to my arms, I flex my biceps, I'm shortening the muscle. That's a concentric movement. When I go to bring the muscle back down, I'm not just dropping it, my muscles are so contracted, but the muscles are lengthening even though they are contracted. That downward motion is an eccentric contraction. Okay, so when I do a dumbbell curl, I'm doing both. I'm doing first a concentric and then a eccentric contraction. Okay, and then isometric would be the example where I can't even lift the weight. My muscles is contracting, but I'm not changing size. Therefore, isometric, same size. Here's a visual representation. You can see the isotonic. What's happening? Muscle contracts because I'm able to overcome the load. Okay, and you get shortening of the muscle. Whereas with an isometric, down here, okay, you see the muscle. Am I moving? Am I changing, moving any load? No, but the muscle is still contracting. Because remember, there are connective tissue portions. There are elastic components of the muscle. So you're contracting the muscles are contracting. What you're doing is stretching those elastic components of the muscle. Okay, and you can see basically the general, looking at 1, okay, muscle at rest. At 2, what do you see? 2 is an isometric. So are we changing the length? Not changing the length, but what are we doing? The muscles are contracting, but what's being stretched is just the elastic component of the muscle. Okay. With isotonic, we do stretch the elastic component, but we're also able to overcome the load, so we're able to contract the muscles even further, and therefore we're able to get shortening, and we get our isotonic contraction. So let's look what's going to vary the strength of the muscle contraction. Okay, because when I lift one muscle, a 20-pound dumbbell compared to an 80-pound dumbbell, I have to create better strength. So how do I do that? I'm using the same muscles. Okay, we can influence how much the number of muscle fibers, also the thickness. That's usually what you're doing in weight training. You're making the muscles thicker. And another component is the initial length of the muscle fiber also plays into how much strength can be created. So that brings us to the length tension relationship. And basically the simple component is the length tension is at what length will I create greatest tension, and that length is where I can get the most number of cross bridging between the actin and myosin. So you can see going up on this is tension being created. Here being the maximum at C. Okay, that's the greatest tension because there is an optimal resting length for those sarcomeres in my muscles. Okay, what do you see happening because here you can see there's lots of ability of interaction between myosin and actin. Okay, we can get great amount of cross bridging. Therefore, we can create great tension. As we slack the muscle and what's happening is as it gets closer together, there's less room, and also it gets wider, and these myosin and actin get further away from each other. So when we slack it, we're decreasing again the amount and numbers of cross bridging that can occur, and therefore tension decreases. Also, if we go the other direction from optimal, we stretch it. Now you can see, is there as much interaction here with myosin and actin as we're having up here? No, there is less. So there, as we come down this way, there becomes more and more, or sorry, less and less ability to create cross bridging, and therefore our tension decreases. Okay, so that's the length tension relationship. We're going to pull back to this again in cardiac muscles, so it's a good thing to learn because we will be utilizing this again when we start talking cardiac muscle and cardiovascular. So another factor can affect muscle contraction, or the strength of muscle contraction, is size of muscles. Okay, so we cannot increase muscle fibers. You can't actually increase the cells, okay, but you can increase production of actin and myosin. So as you're straining those muscles during weightlifting, you're inducing the muscle to create more actin and myosin, and eventually if you do enough of it, you can actually induce more myofibrils to be produced. So you can basically make that muscle thicker. With more actin, myosin, myofibrils, you can create greater tension. So when you're weight training to get the muscles bigger, you're gaining these more proteins, these actin and myosins, and so you're able to generate more strength. So let's look at what controls the motor unit the muscle contraction, and that's basically the motor unit. The motor unit is a motor neuron, and then all the muscle fibers that one single motor neuron will innervate. So many times one neuron will innervate multiple motor fibers. Okay, and that is what a motor unit consists of. Okay, contraction of the motor unit, the force created, is dependent on the types of muscle fibers, which we'll talk about, and also the numbers of motor units in the muscles. Okay, so how do I get, when I lift a 20-pound muscle compared to an 80-pound muscle, I have to create greater tension. And how can I do that? I can do that by recruiting more motor units. So a muscle's made up of multiple motor units. It's not just one neuron controlling all the muscle fibers in it. There is many. So if I recruit more motor units, I can increase the strength of my contraction. Okay? And as I increase the stimulus, I can activate more and more motor units. Typically, we're going to talk about slow twitch and fast twitch. The low threshold, the ones that would fire, motor units that would fire first, would be those containing the slow switch. And as I continually get more and more rigorous or have to increase my strength, I will activate greater and greater motor units that even have fast twitch muscles. Okay, so to get greater strength, we're going to recruit more motor units and different type of muscles. So those strongest being the fast twitch muscles and the slow twitch being more the posterior muscles. Okay, so as I increase strength, I'm increasing more motor units, but also the type of muscle fibers that are generated or being contracted to produce the force. So now let's look at muscle subtypes. There's 3 subtypes. We got the slow twitch. Those are going to be mainly posterior muscles. They are oxidative. Okay, so they use oxidative phosphorylation. We got the fast twitch oxidative glycolytic, so they can use both oxidative phosphorylation and glycolysis, and then the fast twitch. Okay, and you can tell down here, we can see the difference in actually production of tension. A would be the fast twitch glycolytic. B would be the fast twitch oxidative glycolytic, and then the slow twitch would be C here. You can see the production of force is much slower. So that's kind of one of the key characteristics of these muscles. Okay, but also the energy they use is a key component, because it will come with usually the faster the twitch, the less fatigue resistant it is. Looking at some characteristics of the muscles, you can see the fast twitch have a greater diameter, where the slow twitch are smaller diameter, so produce less force. Myoglobin has to do with oxygen levels. It's a reserve for oxygen. Since the fast twitch only use glycolysis, they don't need oxygen, and so therefore they have little to no myoglobin level, where the slow twitch is all oxidative, and so they have a high level of myoglobin because they need the oxygen to produce energy. And capillary density, that allows us to bring oxygen in, right? And where do we need oxygen? We need it for our type 1, not our type 2X, because these guys are glycolytic. So that goes along with the capillary density. Kind of the things I want you to know between the differences between the muscle fibers is mainly, okay, the speed, okay, and also the endurance. Okay, because which one's more fatigue resistant, right? When we look at a sprinter, they're using the fast twitch because they're generating great forces, but they're not able to do that for a long period because those fast twitch muscles are easily fatigued. So now we're briefly going to touch on the cardiac muscle and the smooth muscle. You can see, right, they're striated and they have the intercalated disc. Basically what we're going to talk about here is what's happening at the intercalated disc. You got the desmosins and the gap junctions, which we will talk about in greater detail when we get to cardiac muscle. But I want to talk about the EC coupling. So basically, it's very similar. We create an action potential, but if you remember before on the skeletal muscle, the action potential came down and hit those DHP receptors. In this case, we got the voltage-gated calcium channels, and those are going to open and allow extracellular calcium to be brought into the cell. And that calcium increase is actually going to cause the release of calcium from the sarcoplasmic reticulum. So this is what we call calcium-induced calcium release. Okay, so it's a little bit different, Most characteristics are the same because they have sarcomeres, but the difference is the EC coupling, and it's not DHP receptor, it's calcium-induced calcium release. And finally, smooth muscle, you can see they don't have sarcomeres. They're arranged in this oblique type pattern. So when they contract, they make kind of like this booger type muscle. Okay, they bring them in almost like a bag of muscle. Okay, and then there are two ways to stimulate it. You got a single unit and a multi-unit. Single unit, these muscles have gap junctions, so we can just stimulate this muscle fiber and you get stimulation of these other muscle fibers. Okay, where a multi-unit, this is less. Okay, this is the more common, the single unit. The multi-unit is the less common, but you can see here we have to activate each of these muscles individually since there are no gap junctions between them. And then finally, here comes the coup de grace, the EC coupling for smooth muscle. For our cardiac and our skeletal muscle, we had troponin and tropomyosin as our regulators. Here, it's going to be phosphorylation of the light chain, the myosin light chain. So phosphorylation is what's going to cue whether the myosin head is on or off. Okay, and how that is done is, again, we have an action potential. Voltage-gated calcium channels are open, but in this case, the calcium binds to the calmodulin complex, and that causes MLCK to be dephosphorylated. This is an enzyme, myosin light chain kinase. So it's going to phosphorylate the myosin light chain. Okay, so it gets dephosphorylated. Now it is active and it's going to go phosphorylate this myosin light chain. Now we have that phosphate. Okay, this is going from here to here. We get that phosphate, and now we can get cross bridging. Now the myosin is active and will cross bridge. Okay, without this phosphate, it does not cross bridge. So we have a second enzyme to cause relaxation. We got a myosin light chain phosphatase. All right, when it's active, it's going to remove the phosphate, and now the myosin light chain is inactive and will not cross bridge. All right, and so that's it for smooth muscle and muscle in general. Thank you very much.